Our next interviewee will be Professor Eamon Quigley, formerly of APC Microbiome Ireland and now head of digestive disease and hepatology at Houston Methodist Hospital in Houston, Texas. He will speak on the topic of probiotics and their relationship to live biotherapeutic products.
One of the good things here is that the definition has remained fairly consistent over the years. And I think most people would now accept a probiotic as being a live organism that, when given in adequate amounts, actually confers a health benefit to the person who takes it. So there are three critical elements to that: live, adequate amounts, and health benefit. And unfortunately, if you don't satisfy those three requirements, you really shouldn't be calling yourself a probiotic. That's not the case, unfortunately. The term live biotherapeutic was really recently introduced by the Food and Drug Administration in the US to describe a particular category, namely microbes or their products which might be used as a drug. Because probiotics in the US, and it's just a regulatory thing, are classified as food supplements or food additives, etc. So they're not regulated the same way as drugs, but the live biotherapeutics really bring you into the drug category, and you should be regulated as such. Unfortunately, that's the case, and it's very confusing for the lay public, particularly when they look at their shelves, because there's stuff on there which is not live, or certainly isn't live for the duration of its shelf life. It's never been tested in humans, so it's never been shown to provide a health benefit, and they've never worked out the dose, so they don't know whether the amount is adequate or not. So it's very confusing for the public. Absolutely, and this is another myth that's unfortunately promulgated by even ourselves, because we write reviews and we refer to probiotics as if they're all the same. One probiotic is the same as the other, which of course is not true. And there's been a lot of nice science which has shown that even a very small change in the genome of a probiotic can completely change its function. So we've got to be very specific when we talk about probiotics about the specific strain and also the specific use for which it's intended. That's a big problem. In fact, right now at this moment, I'm involved in a review which is attempting to address how one should do meta-analysis in probiotics, and that's one of the points we're trying to make. Yes, there are some instances where probiotic effects may be somewhat generic, but a lot of the time they're not generic. Actually, they're highly different and they're highly heterogeneous. So you've got to be very careful about grouping probiotics in that manner, which you can come up with really misleading conclusions. Well, I think I would like to mention a few. I think there is good evidence, for example, that probiotics taken with an antibiotic in somebody who's getting an antibiotic for whatever can reduce the risk of getting antibiotic-associated diarrhea or even C. difficile colitis. So I think that's one good example. There is good evidence in a much more severe condition, namely necrotizing enterocolitis, which is a devastating condition that occurs in premature infants in particular, that their probiotics can prevent the development of this condition and even reduce the risk of death. And I would also suggest our own evidence, which is in irritable bowel syndrome, that a very specific probiotic can actually reduce the symptoms of that common and sometimes disabling condition. I think that would make life easier. Unfortunately, I think we're stuck with the term probiotic. I think it's going to be very hard to shift it. I've been thinking about this quite a lot recently, and I think ideally we should be talking the same way we talk about an antihypertensive or an anticoagulant or an antibiotic. We should be talking about more specific terms. But I think we're stuck with probiotic, and it's got problems for a whole variety of reasons, but I think we're stuck with it for now. I think the advice is fairly simple. First of all, you should look at the package and see what it says. What does it say it contains? If it doesn't even specify what it contains, you're in trouble. Then does it actually give the number of bacteria that are supposed to be in there? That's a reasonable indicator. And then does it tell you anything about how that has been tested? So what you want is you want a probiotic which has been tested adequately to make sure what's in there and that nothing else is in there. Secondly, that they've shown that it's viable, it's alive for the shelf life of the organism. And thirdly, they got some information on what benefits it may have or not have. Unfortunately, that takes a bit of work. You won't find that all that on the package. The simple answer is that you should trust reputable producers. And that may sound like a fairly simplistic approach, but I think that's about as good as we can get right. Absolutely. I think particularly more clinical trials. And, it has been shown over the years that if you do good clinical trials, you actually can show benefit from probiotics. A lot of trials are not done for a very interesting reason, because probiotics are largely produced by food or supplement companies which do not have the budgets for clinical trials that the pharmaceutical companies have, and they're not required to do them. So why do them? Because all you get is negative results. So there has been a real shortage of high quality clinical trials in this area, but they can be done and they can produce positive results, and that can have an impact. This is one of the issues actually that have been pointed out over the years, including by people like yourself, that there's a deluge of studies showing that every probiotic does everything in animal models, but few of those have translated into humans. Now, as well as clinical trials, there are other things you can do in humans, and some of the studies we've done ourselves, where you can see that it does something that could be of benefit. For example, it might modify an inflammatory response. It might modify a behavioral response. These are important indicators of potential benefit. Those studies are also valuable.